So, good morning everyone. Welcome to Tamali's classes. In today's topic, we'll be talking about axis and pattern formation in Drosophila. This topic is very important for CSIR-NET life science. So, let's get on with it. Okay. So, first of all, if you see the structure of a Drosophila, okay. So, have you seen the structure of Drosophila? So, you know that there's a head part out here. Okay. There's a head part out here. And then you have the body part, thorax, you can say, then the body part. And then you have the anterior part. So out here, there are two axes, okay? See. So this is known as the dorsal and ventral axis. This is the anterior and posterior axis, okay? So in this topic, we'll be talking about this axis formation. So how anterior posterior axis is being formed and how dorsal and ventral axis is being formed, okay? How the patterning is occurring. So starting with it, so first of all, we should know that this step is occurring in the oogenesis part, okay? So there are some maternal genes involved out here. So what are those genes? We'll talk about it later in my future slides. Okay, so there are some key genes involved in the development of Drosophila. So I'll just mention those genes. So you know there are two types of genes out here: maternal effect genes and the zygotic genes. So maternal genes will form the zygotic genes, and then they will lead to the further patterning and the formation of different parts of the Drosophila. So here we have this egg polarity genes, gap genes, pair rule genes, segment polarity genes. Okay, so we'll talk about it later. So let's see how it's occurring. Okay, so just this one, I'll just tell you the basic. I'll tell you how to see the maternal effect genes. So what is happening? These maternal effect genes, they are forming these gap genes. Okay, they will activate the transcription of gap genes. These gap genes will form the embryo part, that is the head, thorax, and abdomen part. Okay, then also they will activate the transcription of pair rule genes. These pair rule genes, what they will do? They will lead to 7 wide segments. Okay, they will form the 7 wide segments, and then this will activate the transcription of segment polarity genes, which will again form the 14 segments, okay, in the embryo. And lastly, they will activate the transcription of homeotic selector genes. So remember this gap, pair rule, and the segment polarity. These three together, they will activate these homeotic genes, which will further lead to the formation of unique segment, okay, unique segment identities. I have mentioned here homeotic genes, what they will do. Like suppose the antenna is transformed into legs part, or you can see replacing the antenna with leg part, okay. Then also about the generating the eyes on wings, legs, and antenna. So there are some unique segments which are formed by this your homeotic genes, okay. So some unique segments, they are formed by this homeotic selector genes. So coming back to the anterior posterior axis part and the dorsal ventral axis patterning part, okay. So this we have understood that there are the axis dorsal ventral, anterior posterior. Now first we'll talk about the anterior posterior axis, okay. So to understand this, it's not I hope you will understand, like go through it slowly. Let's start, okay. So now in a cell, okay, suppose this is the embryo out here, okay. So see out here, the initial part, the beginning part, these are all nerve cells, okay. The first part, they're all nerve cells, okay. And this is the uncommitted part, follicular cells. So out here, this is the follicular cells, okay. This is the follicular cells. So first we'll talk about the posterior part, okay. So in posterior part, this is the oocyte, and here this area has been focused in this diagram. You can see here. So what happens in the follicular cells? There are some receptors present, okay, known as the torpedo receptors. So they are present out here. They look like this. You can see out of the orange one, orange structures. So these torpedo receptors, what they do, they will accept the gherkin proteins, okay. So what happens in nucleus? In the nucleus, they will form the gherkin mRNAs. This gherkin mRNAs are accepted by these torpedo receptors, okay, and it leads to further processes like formation of microtubules. You can see out here, it leads to formation of microtubules, okay, this first one. Then the second one, you can see out of the structure. I'll just show you in the structure. See, the microtubules are formed, okay. So there are two ends. One is the negative end and this is the positive end. So there is a dynein protein, okay. What this dynein does is it will bind with the bicoid mRNA. So let me tell you one thing first. See, there is bicoid mRNA, there is nanos mRNA, and there is hunchback and caudal. Okay, there are these four type of mRNAs, okay, present out here. So basically, in a cell, okay, in this part, what is happening is, I'll show you. See, in this part, what is happening is out here that your bicoid is towards the anterior part, okay, and nanos is towards the posterior part. So that means, what did I mean by this? That in this part there is nanos, and this part there is anterior part, there is bicoid. Okay, so but the other ones is the hunchback and the caudal. Okay, these are all lying in the center. Okay, these are all lying. They're all lying like they are distributed in the entire uh, entire oocyte, you can say. Okay, so they are not distributed right now. So what's happening first of all is that this dynein, see the second picture out here, this dynein, this red color you can see, this dynein, what it's doing is it is binding with the bicoid mRNA and carrying it towards the negative side. Okay, similarly out here we can see in the this part, in the lower part, you can see Oscar mRNAs. So Oscar mRNAs and kinesin 1, they are moving towards the positive part. Okay, so while this dynein and okay, I'll show you out here, dynein and bicoid mRNA, while they are moving towards the left hand side, towards the negative part, in the below you can see the Oscar mRNAs and the kinesin 1, these two are moving towards the right hand side, and this Oscar mRNAs is binding with the nanos mRNA, and it is associating with it and holding it towards the posterior part. It will not let the formation of hunchback out here. It will only associate with nanos and keep it towards the posterior end. It will not let the nanos to go towards the bicoid. Towards the anterior part. Similarly, even the bicoid cannot come towards the posterior part. Okay, it will stay near the anterior part. Okay, so I hope you understood this one. Now, these torpedo receptors, they are analogous to EGFR in vertebrates, and the gherkin proteins, they are also analogous to TGF alpha. Okay, so this you should remember for exam. Now, coming to the graph part, where I will tell you this in brief. I will explain this to you what is happening actually. See, I've told you that on the posterior part. See, on the posterior part, I told you. Okay, now coming to this part. Okay, in the posterior part, I told you that there is nanos out here, right? And I told you there is bicoid. Okay, and it's somewhere like in between, all over. What is lying out here? Hunchback and caudal, I told you. Okay, so what's happening out here? See, now bicoid, what will happen? Bicoid will not let caudal to express, okay, caudal protein to be formed. That means it will not lead to the translation of caudal protein. Okay, or you can say caudal mRNA will not be translated to caudal protein. By whom? Because of bicoid. It will inhibit that. Similarly, nanos at the posterior end will also inhibit the translation of hunchback mRNA to hunchback protein. Okay, so now this much you've understood. Now see the graph part. So I told you in the graph part, if you see the anterior portion, this is anterior, this is posterior. In anterior, if you see the diagram out here, okay, see out here in the anterior part, the bicoid is more. Okay, and as soon as it moves on towards the posterior part, the bicoid concentration decreases. Similarly, in the posterior part, okay, out here, the nanos part is more, and as soon as it moves on towards the left hand side, the concentration decreases. Okay, but this hunchback and caudal, they are out here present. This is the graphical presentation of the mRNAs. Okay, when it's in mRNA form. So bicoid mRNA is more in anterior, and nanos mRNA is more in the posterior part. Okay, now when they are forming this proteins, okay, so you can say cleavage embryo proteins. So the second diagram out here, you can see what is happening out here. Bicoid, since I told you bicoid will inhibit this caudal, and it will help in the hunchback translation. Okay, so out here you can see hunchback is forming. Okay, and out what will happen? Nanos will inhibit the hunchback mRNA translation, but help in the caudal formation. Okay, so it's a caudal formation. Understood? Okay, so this is all about your anterior posterior axis formation out here. So this is some just details I've given out here. So I told you that there are anterior, posterior, and also terminal sets of egg quality genes. Okay, so Bicoid I told you, nanos I have told you. So that there's also a third signal, okay, that is generated symmetrically at both end of the egg, that is known as tyrosine kinase receptor called as torso. Okay, so now this torso will help in the formation of this head and tail, okay, and also the like it will determine the future gut, okay, this torso part. Now moving on to the dorsal and ventral patterning in oocyte. Now what is happening in this part? Let's see. Here I have told you that what is happening. Here I have told you that about Gurkin protein I have mentioned to you, right? So I'll just mention to you a bit in more in details. Okay. See, this Gurkin mRNA I told you it's forming, it's coming out from this nucleus. Okay. From here, Gurkin protein. Okay, let's take a new slide out here. Let's explain it out here. It will be helpful for you also. Now, see here. I told you in this oocyte out here, this nucleus is forming this mRNA. Okay, Gurkin mRNA. So this Gurkin mRNA, they will form Gurkin proteins. Okay, this Gurkin proteins, they will go and bind to the anterior part. Okay, or you can say the dorsal part out here. So what will happen? They will first, okay, just imagine they'll go to the anterior part. So out here there are some torpedo receptors where they will go and bind. Okay, they will go and bind. Remember, this binding of Gurkin mRNA, Gurkin proteins with the torpedo receptors will inhibit the formation of pipe synthesis. Okay, because pipe synthesis is very essential for the formation of dorsal and ventral patterning. Fine. So for dorsal and ventral patterning, this pipe synthesis is important, and especially in the ventral. For ventral patterning, pipe synthesis has to occur. But this pipe synthesis will only occur when there is no binding of Gurkin proteins with the torpedo receptors. Now, since out here, see in the dorsal part, or not dorsal part, you can see out here, okay, take it as dorsal part or anterior part, as I've told in my previous slide out here, Gurkin proteins are binding with the torpedo receptors. Okay, so that means there will be no pipe synthesis. But in ventral part, now coming to the ventral part, okay, let's see out here. See the structure out here. I'll just explain you the structure. It'll be more helpful rather than reading all the lines. Okay, so if you see the structure out here, this is the oocyte that I've shown you, the nerve cells out here, and you know the follicular cells are present. Now, in this oocyte place, I'll show you. See, this is the oocyte. Okay, this is the nucleus out here. This is the oocyte. So this is the vitelline membrane. Okay, and this is in the center. This is the perivitelline space. Okay. So what is happening out here? There are some toll receptors present out here in the oocyte. Okay, there are some toll receptors present out here. I think the structure doesn't have that. Okay, here there is. So you can see there are some toll receptors present out here. You can see this thing, toll receptors. Okay, this pink ones. Okay, so what happens inside this? Okay, inside this membrane, what is happening here? First of all, I told you that Gurkin proteins are being formed by mRNA. Okay, so Gurkin proteins will go and bind with the torpedo receptors. So you can see this green color. Okay, this green out here you can see. Okay, so these are all pipe synthesis. Okay, so now Gurkin proteins will go and, okay, this first diagram, it shows you that the Gurkin proteins are binding on the dorsal part. You can see it's going up, okay, upwards, and is binding with the torpedo receptors present in the follicle cells. Okay, now come down. Now see how Gurkin proteins are binding with the torpedo receptors. Okay, so when they bind with the torpedo receptors, they form the pipe synthesis. Okay, so here what is happening? The pipes are being formed. Okay, the pipe synthesis are being formed. So they are binding with the torpedo receptors. I'll show you. Okay, see, T is for torpedo, this is for Gurkin proteins. Okay, so T and G is binding out here. Okay, so the structure out here. Okay, so T and G, they're binding out here. So when this T and G bind, when the Gurkin proteins bind with the torpedo receptors, what's happening? You know, there are some vitelline proteins out here present. So they get sulfated. Okay, sulfur groups are added, so they get sulfated. So sulfation of vitelline proteins occurs. This will recruit GD. GD is gastration defective. Okay, so now see here. So, okay, pipe synthesis and torpedo. This will lead to formation of the sulfation of between proteins. Okay, sulfation of between proteins out here. So this will recruit GD. Okay, this will recruit GD or gastration defective proteins out here. Now when this will happen, with this GD, what will happen? Snake protein will bind. Okay, snake protein, which is in inactive state. Fine. So I'll just show you. So TP, then sulfation of proteins, and then GD will come and bind. With GD, snake protein will come and bind. Okay, which is inactive. Then this will become active, and again what will happen? Ester will come and bind. Ester protein. Okay, then Ester. Ester will come and bind. So this is also inactive. This will again become active. So once ester becomes active, what will happen? It lead to formation of spatzel. Okay. Then again, lead to formation of spatzel. Okay, out here you can see that this green out here is spatzel. Okay, forget the diagram. Just look at what I'm showing you. Okay. So spatzel will lead to formation of inactive spatzel. Okay. So now this inactive spatzel, what it will go? It will go and bind to the toll. Remember I told you there are some toll receptors out here. See out here, I told you now in the, see this part in the oocyte membrane there are some toll receptors. Okay, toll means it's like a gate. वो gate पे खड़ा है. So it's stopping the movement of any other proteins. So what will happen? This spatzel will get activated and it will go and bind with this toll. So once spatzel gets bind with this toll out here, see in upper part, see in upper part. So spatzel, when it goes and bind with the toll, what will happen? It will lead to formation of not formation. It will lead to binding of another proteins known as tube and pele. Okay, tube and pele, T and P. Okay, so now here you can see tube pele is bound with the spatzel and the toll receptors. Okay, when this binding occurs, what will happen? This pele out here, what is the pele's function? It is the it will phosphorylate. Okay, what will it phosphorylate? So remember, inside this there is a complex of cactus and dorsal mRNA. Okay, mRNA dorsal protein. Okay, it's a dorsal protein also. So this dorsal protein and cactus, there is a complex. It is present inside the oocyte. So what will happen? For this ventralization to occur, for the dorsal ventral patterning to occur, this C and D, this cactus and this dorsal, they have to separate. For their separation, it's important that toll and pele has to be, they has to come and bind together into a complex, and pele has to phosphorylate cactus. Okay, so after this this tube and pele binds together, this pele becomes activated, and pele will go, because earlier pele is inactive, is in inactivated form. So when tube and pele binds, so pele will become activated. It will go and phosphorylate cactus, leading to separation of cactus from the dorsal protein. Okay, so now we have cactus separate and we have dorsal separate. So when this occurs, cactus will generally, like gradually it will leave the cell out here, and dorsal will move into the nucleus. Okay, so dorsal will go into the nucleus. As soon as dorsal goes into the nucleus, what will happen? Dorsal will lead, see, when dorsal moves into the nucleus, it will lead to the ventralization of the cells. Because see, for ventral patterning to occur, the dorsal has to move on to the nucleus part. Okay, so now dorsal will go to the nucleus part. Pipe synthesis has occurred in the ventral part, so it will lead to the ventral axis formation. Now, since there is no, since Gherkin proteins is present in the dorsal part, so out here what will happen? There will be no pipe synthesis, and it will lead to the dorsal axis formation. But since in the ventral part the Gherkin was not present, so pipe synthesis occurred. Okay, and it led to the ventral part. That means you can say the dorsal protein out here is acting as a initiator. You can say it is helping in the ventral axis patterning or formation. Okay, now this is what I've explained you till now. So these are type of genes, as I've told you. So gap genes, what does it do? I told you already out here. See what they're doing. So that's why I told you I'll explain it later, because it's not required now right now for explanation, because here you can see all the functions are given for all the genes. Okay, so there's also another called bithorax complex. Okay, bithorax complex, sorry. So bithorax complex, what does it do? They're responsible for the posterior segment diversification. So out here 3 genes are involved. So one is for the ultra bithorax part, and one is the antennapedia complex. Okay, so in antennapedia complex there are 5 genes: labial, proboscipedia, 6-comb reduced, deformed, and antennapedia. So these genes are responsible for the head segments, then ANTP and SCR. Okay, so these so remember just head segments. Okay, antennapedia complex is responsible for the head complex. Fine. Now ultrapythorax region is for the thorax part. Okay, they will identify the abdominal segment. Fine. So and will also result in the transformation of wings and flies. Okay, sorry, wings in the flies. Okay, now this is done. I've told you about segmentation genes. So I told you about the homeotic genes also. So why is Drosophila used as an experimental organism? Because it's easy to use them. Okay, they are cheap to breed. Okay, they are rapid in the reproductive cycle, and also out here the genetic redundancy is less. Fine. And also the Drosophila, the patterning, the genes that control the patterning of Drosophila, they are having close counterparts in the higher animals. We have already seen the torpedo and the gherkin protein out here, right? So this is all about the axis formation in Drosophila. I hope you have liked it. If you have liked my class, do click on the like icon, share it with your friends, and do subscribe to my channel for more such videos. And all the best for your CSI exam, and keep learning. Thank you so much.